So we're going to get into a pretty heavy unit, a pretty heavy topic when it comes to this course, and that is metabolism and cellular respiration. Right? Because what we're going to go into is we're going to basically talk about, on a cellular level, the physiology of how cells get their energy, how we harvest energy from the foods we eat, and then convert them in a form that our cells can readily utilize to power all of these other reactions that are required. So this first one, energy metabolism, I'm actually going to split into two separate videos. And so we're going to go through a few things. I'm going to go through the whole list here since we won't see this slide again. But some of these things will be covered in this video and others in the next. But we're going to talk about catabolic and anabolic pathways and how those are coupled, and that we have energetic considerations for each. We're going to explain the mechanisms that ATP is synthesized in humans. We're going to talk about the 4 basic steps of cellular respiration. We're going to talk about the role of the redox reaction and coenzymes when we're producing ATP. And then we'll get into some of the nitty-gritty with glycolysis, the transition reaction, Krebs cycle, also called the citric acid cycle, and the ETC, or the electron transport chain. Talk about them, and then get into kind of what's happening in the cell, the reactants, the products, to how much ATP, and then how many electrons basically are generated by this through NADH and FADH2. We'll also talk about some other pathways that can be used, like fermentation and when that occurs and how much ATP we get out of that. And then talk about the Cori cycle to convert lactic acid to glucose or glycogen in the liver. Lastly, we'll talk about the role of chemiosmosis in powering ATP synthase, which is really the money maker when it comes to ATP production. So we have to have some reminders. I know we've been hitting chemistry topics a lot in this course, but that's because, again, we're bound by the rules of chemistry, right, as living systems. So some reminders that the chemical bonds that make up all the different molecules in our body, there's energy in those bonds, right? So when we look at like the carbon-carbon bonds that we find in like glucose, like the bonds between 2 carbons, right, within glucose has that ring of 6 carbons. So we have all those bonds between those carbons. There's energy there. And then there's energy in the phosphate bonds in ATP, right? So every chemical bond has some energy in it, but these are the ones we're going to focus on for this class, right? As the carbon-carbon bonds and the phosphate-oxygen bonds in ATP. So in order for us to get this energy from these chemical bonds, we have to break them, right? Catabolic reactions to break them, because when we go through that breaking, it's going to release the energy from those bonds. Think of the chemical bond like a storage facility or a bank, right? And when you break it, right, you're going in and pulling something out of storage, or you're making a withdrawal, right? So we're getting the energy when we break those bonds. That energy, when it's released, can be captured and used to power things like enzymes and other reactions that require that input of energy. So you can kind of see how it's usually paired, right? You can see how there's this pairing basically that can occur. This energy transfer is normally, again, achieved by the redox reaction, which again is a paired or a coupled reaction, which helps us with forming ATP. So let's get to know ATP a little better. So ATP, or adenosine triphosphate, is kind of the energy currency molecule in the body, right, in living things. And it has this nice structure where it has an adenine base, like nitrogenous base. It has a sugar, and then it has this chain of 3 phosphates in a row. And as you go down the chain, from the first to the second to the third phosphate, there's very high energy amounts in those bonds, right? Especially as you go down the line. So if we're looking here in the picture, we see phosphate 1, 2, and 3, and the bond between the second and the third, tons of energy there. Tons of energy between those bonds. So high energy, we want to be able to harness that. So we can go through phosphorylation, which phosphorylation is our process to build ATP back up. So when we use ATP, we just break off a phosphate, right? So we go from ATP, adenosine triphosphate, to ADP, or adenosine diphosphate, plus the phosphate that's free floating. So phosphorylation is the process of building that ATP molecule, right? And because we have that energy bond, or that bond between the second and the third phosphate is so high energy, it's going to require a bunch of energy to build that bond back up. It's not just going to happen on its own. Dephosphorylation, we're breaking it down. We're releasing the energy between those two phosphates, right? We're breaking the bond. And that's going to release that energy, which we can harness and use to power other chemical reactions. So ATP is kind of like an energy currency molecule. It's helping kind of harvest energy, right? Collect that energy up from somewhere else through the process of building ATP, and then we can break it down to release that energy elsewhere where it's needed. So we can see that ATP is used with these hydrolysis and dehydration synthesis reactions. So let's say we're looking at some simple molecules like glucose and whatever else is listed here as well. We can have ATP, break ATP down, and the release of that energy from ATP can give us what we need to go through an anabolic reaction, right? Transferring the energy from the ATP to these simple molecules so that they can build up to something more complicated. And then when we need to break down those complicated molecules, right, we can break them down and use that breaking down process to produce the ATP back again. So you see the ATP is kind of recyclable, right? We're going around and around and around, and one single ATP molecule can do this 1,000+ times a day. So there's a coupling, right, between anabolic and catabolic reactions where we're transferring energy from one reaction to the other. Remember that anabolic reactions require energy input because they're endergonic, so they need ATP in order to power them. So something like a dehydration synthesis reaction where we have to get the input of energy. Catabolic reactions we can use to create the ATP, to build ATP. These are exergonic, they release the energy, and that energy can be harvested and used to reform that phosphate bond with that last phosphate group. So remember that catabolic we're breaking down, and so hydrolysis reactions would be used to break those bonds. Energy is released and harvested to produce the ATP. So energy metabolism is basically just we're looking at mainly the production of ATP, right, by breaking down things like glucose, glycogen, fats, and proteins. So we have a couple different places we can get our fuel, right, that we're using to produce ATP, depending on what we're doing and when we last ate, and depending on, what we've already depleted, right, will tell us what we're going to use. So the first thing we go for is the glucose in the blood, right? That's going to be the first thing we go for. But if that has been depleted to some degree, then we can start breaking down the glycogen in the muscle and the liver. That's kind of like our checking account, right? Like you use your debit card to go pay for something. And if you deplete that down, you can go into your savings account, right? So this would be your lipids, which would be your fats right underneath your skin and your abdominal fats. And then if absolutely necessary, right, this is emergency funds at the very end here in red, absolutely necessary, you've depleted the glycogen, you've depleted the lipids, you need to go for the proteins in the muscles, which is a last resort. So in people, ATP can be generated in two ways, right? We can either basically directly go from ADP to ATP. We're literally just slapping that phosphate on. This is called substrate level phosphorylation. Or we go through oxidative phosphorylation, where we're indirectly creating that ATP because we're doing an electron change. So the way that you can remember these, honestly, is that substrate level phosphorylation is what's happening in steps like glycolysis and the Krebs cycle. Whereas oxidative phosphorylation, this is what the electron transport chain is taking care of, right? And we'll get into some details to some degree with these. I'm not so worried about you remembering these. I'm more worried about talking about the actual process of cell respiration and the key moments that happen in that process. So if we're looking at the most effective mechanism of making ATP, because there's multiple ways that we can get there, right? ATP can be made in several different ways. We can use fermentation, we can use cellular respiration, right? We can use anaerobic respiration, right? There's a few different pathways, but they're not all created equal, and they all have their pros and cons. But the one that's really most effective and most common, especially when we're looking at eukaryotic cells that have mitochondria, is cellular respiration. This is also known as aerobic respiration because it requires oxygen at the end. So we're basically going to start with glucose, and we're going to break glucose down, and in that process produce ATP. And so right there in the middle we see our general equation, right, that shows us what cellular respiration is doing. So here's our reactants. We start with the glucose, we have an input of oxygen, and by the time we get to the end, we end up with some carbon dioxide, some water, and some ATP, right? These CO2 and the H2O, both of which are kind of the byproducts. The ATP is what we want, and we're getting about 36 to 38. So for one single glucose, we're getting a good amount of ATP in this process. But the whole thing, we notice we're breaking those carbon-carbon bonds, right? We go from a molecule that has 6 carbons bound together to 6 individual carbon dioxides where we have just 1 carbon each. And so it's that process of breaking the carbon-carbon bonds that allows us to do this. Again, we can directly produce this ATP with substrate level phosphorylation, or the indirect, where we're using the electron chains to go through and make ATP through oxidative phosphorylation. Now, we've mentioned the word metabolism over and over and over in this class, and remember that's just the sum of all the chemical reactions. But when we're looking at energy metabolism, a lot of the reactions that are occurring are paired, right? And they're redox reactions. Remember redox, or if you remember oil rig, I wrote that on the board, but oil rig, right? Oxidized is losing and reduced is gaining, right? So when we're talking about what is losing and gaining, we're talking about electrons. Now, because we're talking about the redox reactions and losing and gaining electrons in a biological system, something has to carry those electrons somewhere else, and that's where the hydrogen ion or the proton comes in. So the hydrogen ion is kind of the shuttle bus for those electrons. So you'll notice that we see hydrogen go along with this. So if you see the hydrogen, you know that we've already been reduced. If the hydrogen's not there, then you know we're oxidized. And so we see that here. We see our lactic acid, it gets reduced, right? Whereas NAD plus gets oxidized right here. But when we transfer lactic acid to pyruvic acid is oxidized, and NAD plus is now NADH, right? That has been reduced. And we can go back and forth, right, between those. Especially over here, the NAD plus and NADH, we can go back and forth, reducing NAD plus to NADH, and then oxidizing it back to NAD plus. So the glucose, when it breaks down, right, these products, these get oxidized. The hydrogen ion, the proton shuttle bus, and the electrons are going to be transferred to a coenzyme, right, which is again our NAD plus or our FAD, right, and those are going to be the shuttle buses, right, to carry those electrons elsewhere. The whole point here, and we'll talk about as we go through the steps, so if this doesn't make sense right now, that's okay, right, we'll be able to apply it in the next video, and it'll make more sense there, that we are basically stripping these electrons, right, in a way to pull them to another step, right, elsewhere. Right, so these coenzymes, the NAD plus, the FAD, are going to be the shuttle buses to move these electrons, as well as the proton that they're attached with, over to the ETC.